Welcome to Concept in Medicine. In today's tutorial, we are going to be looking at the mechanism of bilirubin synthesis. Let's start off by looking at the sources of bilirubin. So with the sources of bilirubin, the main source of bilirubin is by the destruction of senescent red blood cells and prematurely destroyed erythroid cells in the bone marrow. And for that source, it constitutes about 80%. The remainder 20% is going to come from the cellular turnover of heme containing proteins. Then the question is, what are the heme containing proteins? The heme containing proteins, from the word, they contain heme, and examples of them we can easily remember using the PMC square T. And for the P, we are talking about peroxidase, M, myoglobin, the first C, catalase, the other C, we are talking about cytochrome. And for the cytochrome, there are two main types that are highly concentrated with heme. That is the cytochrome P450 and cytochrome C. And finally, tryptophan pyrrolase. So these 5 proteins are known to contain heme. Alright, let's move on and look at how the synthesis of bilirubin takes place. Alright, so first of all, you should know that hemoglobin is made up of heme and globin. When hemoglobin is broken down, you have the globin being sent back to the liver because it's a protein, so that the liver can use it in the process of what? Protein synthesis. The heme will go into the bilirubin synthesis pathway, and with that, the heme, which we also refer to as the heme moiety, is made up of 4 pyrrole rings. 1, 2, 3, 4. These are the pyrrole rings with a central ion atom, as we can see here, and the central ion atom is linked to the pyrrole rings by the methyl bridges. The methyl bridges. Now, if you have the heme acted upon by an enzyme we call the heme oxygenase, and for heme oxygenase, it is produced by the reticuloendothelial cells of the Kappa cells of the liver. So in that case, the heme oxygenase acts on the heme, oxidizing it, resulting in the breakage of the methyl bridges, producing equimolar quantities of carbon monoxide, thereby liberating the ion in the middle here. The reason why carbon monoxide is produced and not carbon dioxide is because there is an incomplete combustion of the methyl bridges, which contains carbon. The ion that is liberated, that's the ion at the center, the ion atom at the central that is liberated, is then carried by a protein we call transferrin to the liver, where it is stored as ferritin. When the ion is liberated, the remnant compound is referred to as protoporphyrin. That is why this whole structure is known as ion protoporphyrin. Once the ion is liberated, what is left is the protoporphyrin. The protoporphyrin is then converted into a green substance or green pigment we call biliverdin. Once biliverdin is produced, the biliverdin undergoes a reduction reaction with the help of NADPH, that is reduced nicotinamide adenine dinucleotide phosphate, NADPH dependent enzyme known as biliverdin reductase, to produce an orange-yellow pigment referred to as bilirubin. The next thing we'll be talking about is how bilirubin is metabolized in the liver. Let's move ahead and talk about the bilirubin metabolism in the liver. So the unconjugated bilirubin binds to albumin, where it is carried to the liver. Once carried to the liver, the enzyme produced in the liver known as gluconeotransferase, which belongs to a class of enzyme we call uridine diphosphate gluconeotransferase, acts on this unconjugated bilirubin to cause its conjugation in a reaction as follows. We have bilirubin acted upon by gluconyl transferase in the presence of a substrate we call gluconic acid. With the help of the gluconic acid and the gluconic transferase, bilirubin is then conjugated in this reaction, where we'll have the bilirubin unconjugated going in to become bilirubin diglucuronide. So in that case, bilirubin diglucuronide is what we refer to as the conjugated bilirubin or the direct bilirubin. The conjugated bilirubin or direct bilirubin is then excreted into the gut, specifically the duodenum, via bile. The conjugated bilirubin in the gut is then acted upon by beta-glucuronidase, which is released and stored in the branch border cells by anaerobic bacteria, mainly the bacteroids. The beta-glucuronidase acts on the conjugated bilirubin, deconjugating it, that is converting it into an unconjugated bilirubin. Note that unconjugated bilirubin or indirect bilirubin is lipophilic, and the gut is also lipophilic. Conjugated or direct or bilirubin diglucuronide is what? Hydrophilic. So the unconjugated bilirubin is absorbed via the gut, since it is lipophilic, and like for like dissolving like, it is absorbed via the gut into the circulation, where it is carried to the liver for conjugation. And this cycle is what we refer to as the enterohepatic circulation. So when we say enterohepatic circulation, what do we mean by that? For enterohepatic circulation, it refers to the uptake of unconjugated bilirubin by the hepatocytes. The conjugation of these unconjugated bilirubin to give you conjugated bilirubin, the excretion of the conjugated bilirubin into the gut, the deconjugation of the conjugated bilirubin, the absorption of the unconjugated bilirubin via the gut, and the reuptake of the unconjugated bilirubin by the hepatocytes. That is what we refer to as the enterohepatic circulation. I believe we've learned a lot of interesting stuff today. Kindly make sure to subscribe to my channel, like, share, and also comment the next concept you would like to see in my next video. My name is Dr. Del, and this is Concept in Medicine. Bye-bye.